Started. So again, thank you, Tina, for the introduction. Yeah, specifically, I'm going to be talking about hemicelluloses. It's my favorite family of polysaccharides. So I really hope that I can convey to you, why I think they're truly fascinating, not only from a biological perspective, but also from a materials and nutritional perspective. So that's a little bit the two big areas of my talk today. But before that, I wanted to give you a bit of a hint of my background. I'm originally like a chemical engineer from Spain, so that means that I speak very fast. So you will have to bear with that throughout the presentation. But I became interested in polysaccharides when I did my postdoc in Australia. So I spent 2 years in the Center for Nutrition and Food Sciences there, and that is where I started learning about biopolymers and starch. Then I came back to Sweden 2011, and I started slowly to consolidate a research program in my university at KTH in Stockholm. And at KTH, we have a big focus, as in many Nordic countries, of course, in forests. And then is when I shifted a little bit from nutritional polysaccharides to more forestry-based ones. And I really wanted to work with hemicelluloses because there was a lot of people working on cellulose, and I thought, hemicelluloses are very cool and are complex, and I found a very nice challenge. Now, currently, I am professor and head of the division of glycoscience here at KTH. So we have a full division focused on carbohydrate biotechnology, biochemistry, and biology, which is pretty cool, I think. And also, I wanted to highlight that, of course, I couldn't leave the food out of my background since I became very fascinated with it. And I became director of a new center here at KTH called KTH Food as of last year, where we're trying to investigate, well, different aspects of sustainability and health of food systems. And also, since last year, I became a part-time scientist at a plant-based dairy company called Oatly, based in Sweden, where we're making alternatives to dairy products using oats. So that's a little bit my background at the moment. But my research interest is basically fundamental carbohydrate biochemistry. We're really interested in understanding the molecular structure of relevant plant complex carbohydrates, polysaccharides, as I say, mainly hemicelluloses, their molecular structure and their multi-scale assembly in plant cell walls. But also we use this knowledge, this fundamental knowledge, to design different processes, normally using green extraction techniques, but also enzymatic modifications to use these hemicelluloses, these plant polysaccharides, in applications as, for example, biomedical materials or food or functional food ingredients. So you can see the two areas that we'll cover today, like a little bit more fundamental plant polysaccharide biochemistry, but also the applications on towards materials and nutrition. And again, my two areas of interest are food and wood. It sounds kind of funny when you just change one letter and you have both. So yeah. But now I'm gonna dive a little bit on the first part of the lecture on the molecular structure of hemicelluloses and how do they impact supramolecular assembly in plant cell walls. So from a biological perspective, so plant cells are surrounded by a complex layer called the plant cell wall. Has different layers, the primary plant cell wall, the secondary plant cell wall. And the function of this cell wall is to provide, of course, mechanical support to the plant cells, but also defense against pathogens. But it has to have very interesting applications because it has to be flexible. So when the cell grows, it has to be able to accommodate these different forces. And then when the plants die and then they get lignified, then it has to have another support, for example, to conduct water, to make the plants and the trees grow very high. And what is fascinating to me is that is all done using three main components: cellulose microfibrils, hemicelluloses, pectins, of course, and also lignin. So these are the main, of course, components on wood of secondary cell wall. And there's a lot of interest and work being done on cellulose. And here I have a parallelism with football. So cellulose, a lot of interest and a lot of research being done on that. Also on lignin. Recently, of course, trying to utilize this very valuable phenolic or a renewable phenolic resource. But somehow it feels that hemicelluloses are somehow a little bit forgotten in this equation, and hemicelluloses somehow are the ones that modulate the interaction between cellulose and lignin. So they have a very important role, which I really hope I can convey in this lecture today. So a little bit of background about plant hemicelluloses. So they are matrix polysaccharides that are able to interact and interlock cellulose microfibrils. And that is due to their, again, fascinating molecular structure. So hemicelluloses have a wide diversity of molecular structures. So if we start with cellulose, we're all familiar with it. So it's a linear polysaccharide formed by beta-1,4 glycosidic linkages with equatorial configuration. So hemicelluloses have a very similar or the same backbone as cellulose. It's also formed by beta-1,4 linkages in equatorial configuration. But as a difference with cellulose, we have different sugars forming part of this backbone. We have not only glucose, but we also have mannose and xylose in the backbone. But also differently from celluloses, in hemicelluloses, we have different decorations. They can be sugars, they can be neutral sugars, for example, arabinose, galactose, but they can also be acidic sugars, like, for example, glucuronic acids. And also, we find in hemicelluloses normally different chemical modifications, like, for example, acetylation and in some cases also methylation. So what is the function of these different decorations and substitutions? So first of all, we think that the presence of these decorations, they can modulate macromolecular conformation and flexibility. We know that the cellulose chains are very stiff, they're insoluble. While we start adding different decorations and modifications, the polysaccharide chains different their conformation and flexibility. They can tune the hydration. I was mentioning also that cellulose are insoluble. Some of hemicelluloses can become soluble. Solubility is a very complex issue. I will not talk so much about it in this lecture. We've looked at it. But also, it can also tune ionic and pH regulation by precisely having these acidic side chains. It modulates the interactions between cellulose and lignin. I will give you some examples of it in my lecture. They can also prevent enzymatic degradation. And you can think of the way cell walls are constituted with all these different polysaccharides, and specifically hemicelluloses with this wide range of glycosidic linkages. You need basically one enzyme for each glycosidic linkage type. So obviously, it really contributes to making it more difficult to be hydrolyzed. And of course, when we're using hemicelluloses for different applications, for example, as films or gels or whatever we can consider them from, all these different substitutions have an influence on the macroscopic, rheological, tensile, whatever property you can consider. So I just wanted to give you a little bit of a historic perspective. I think in the last 10, 15 years, there has been a lot of progress understanding specifically supramolecular architecture of plant cell walls, and specifically the role of hemicelluloses in that. That's work that has been done in different labs in the world. And the reason for this really nice blooming research in plant cell walls in the last 10, 15 years, I think it comes from three different areas. One is plant biology, plant molecular biology, because now plant biologists have the tools to understand how these different polysaccharides in cell walls, and specifically the hemicelluloses, are synthesized by investigating which genes and which proteins are synthesizing this, and basically being able to tune and regulate these plant cell walls. But not only from that, but also from the contributions from the characterization part, from, for example, mass spectrometry, NMR, and other advanced techniques that really allow us now to look into detail at the molecular and supramolecular architecture of plant cell walls. But also, importantly, the contribution of theoretical chemistry and in silico molecular simulations has actually given us some tools to really investigate these roles. And I'm quite happy to consider myself a multidisciplinary researcher. So I have collaborations with plant biologists, theoretical chemists, and I truly believe that with all these different contributions, we can really reach a much deeper understanding of plant cell walls. And I also wanted to highlight the work done in this regard, trying to understand the sequences or the molecular structure of hemicelluloses done by two researchers that are part of the EPNOE community that I really appreciate very much. So one is Paul Dupree in Cambridge and Maija Tenkanen in Helsinki, because they were one of the first ones trying to look at how the different decorations and substitutions in hemicelluloses were placed. So historically, people believed that hemicelluloses were kind of random polysaccharides, and now we know that that's not true. Hemicelluloses have very specific and very regular structures, and that's determined by the way these hemicelluloses are synthesized by the plants. So just a couple of examples. So work done in Helsinki by Maija, using mass spectrometry, they identified that they could see some regular patterns of even substituted decorations. But then it was the work also done by Paul in Cambridge, really identifying which are the different enzymes that can synthesize hemicelluloses. And they identified different enzymes that really locate decorations like acetylation and glucuronic acid at specific and very regular positions. So this is really a nice contribution to the field. So in my lab, we've been working a lot since I started it in mass spectrometry, because I really think this is a really fantastic technique that allows to look at very fine molecular details in polysaccharides. And what we've done in my lab in the last 8 years is to develop different mass spectrometric pipelines to analyze the molecular structure of different plant polysaccharides. So we draw inspiration from proteomics, which is a very well-established field. And in the case of plant polysaccharides or hemicelluloses, these molecules are very big. You cannot analyze them as such normally in mass spectrometry. So what we did first was to devise a protocol starting by enzymatic hydrolysis of these polysaccharides, making use of the very specific biochemical selectivity of the different carbohydrate active enzymes that we have in our repertoire. So you have here, this is an example for xylan. Again, as I was mentioning before, this xylan molecule is too big to be able to be fed in the mass spec. We use enzymes to cleave these hemicelluloses in smaller oligosaccharides, that these ones can be analyzed by mass spectrometry. So here, as you see the pipeline, we create this very broad diversity of oligosaccharides. We separate them with chromatography, hoping that we can get separation of the different isomers. We can even, using the mass spec, select specific molecular masses of the different oligosaccharides. Then we can fragment them using electrons or difference of charges, and then based on this fragmentation pattern, we can really sequence the specific structure of the hemicelluloses. And we've been applying that to basically all possible hemicelluloses, like xyloglucans, mannans, xylans, beta-glucans, etc. But today I will focus mainly on xylans and a little bit on mannans following this approach. And I also, very important to highlight, extraction. Of course, you need to isolate these hemicelluloses from wood or from the different plant tissues. And here extraction is very important. People have been using normally alkali to extract hemicelluloses, but different groups are now using what we call subcritical water or pressurized hot water extraction. And this is a very nice technique that I've been working with also. And it's very nice because it can isolate hemicelluloses, not only in polymeric form, but also keeping some of the functional groups intact that, for example, alkaline extraction does not preserve. So long story short, we've been working on different xylans from hardwoods, for example, on birch, also from softwoods, from spruce. And the idea for this project was, okay, are there different sequences that are regular in the molecular structure of spruce arabinoglucuronoxylan? So this is the molecular structure of the xylan in spruce. You have this backbone again of beta-1,4 xylose with decorations of glucuronic acid and arabinose. So what we did in this study is we used a specific xylanase enzyme that can really recognize where the glucuronic acid is in the backbone. It cleaves just one unit next to it. You can see here the arrows when the different when the enzyme can cleave, and then it releases these oligosaccharides. So what we did in this study is actually to identify that there were specific motifs very well conserved in the structure of spruce xylan. You can see all these different motifs that always share this even spacing. You have the glucuronic acid here, and always 2 or 4 units next to it, you have the arabinose sitting next to it. And then if you start thinking about the conformation of hemicellulose and cellulose, you know, this beta-1,4 linkage makes that you have the C6 acting in opposite now, up, down, up, down, up, down. So if you really keep this even spacing, that means that all the decorations will be facing the same side of the chain if you think of a flat surface. Then we also found a very interesting uneven uronic spacing, very little. And this is also, it was also very interesting. We found different regions when you had clustered uronic acids, very conserved. Always have one glucuronic acid and one next to it. Not 2, not 3, not 4, just one next to each other. So then we started working with different, with collaborators working in theoretical chemistry, trying to understand the interactions of these motifs with cellulose surfaces. And we really could see that basically these side chains, what they do is somehow they anchor and destabilize the interaction with cellulose surfaces, with hydroxylic and the hydrophobic surfaces of cellulose. And that worked very well with, as I say, the work that was being done in parallel in different labs, trying to remove these decorations and study in planta what was happening to these substitutions. But also very interesting with lignin, we have the hypothesis, of course, that hemicelluloses are cross-linked with lignin through carbohydrate lignin complexes, and that the uronic acids are actually one of those potential links to lignin. So to have these clustered uronic acid substitutions, we think that they are acting as potential starting points to the cross-linking with lignin in the secondary cell wall. So you have a very nice example where with the same type of polysaccharide, you can modulate interactions with cellulose and with lignin. And also very interesting story. So when we did this study, these genes that are responsible of synthesizing these consecutive glucuronic acids were not known. So when we published this, we talked to Paul Dupree's group in Cambridge, and then they immediately started looking at these glycosyl transferases, and they found them later, which is actually a very nice example of how you can, from the structural point, influence the plant biology. So finding these motifs will always guide the plant biologists in finding the enzymes that are capable of this. So now I will present, this has already been published, but now I will present two cases that we're looking at the moment. So now we're looking at also other species, like in this particular case, aspen. Aspen is a hardwood, and the molecular structure of xylem is a little bit different. We don't have arabinose, we have glucuronic acid, and we have acetylations. And so Pramod, this is work done by this postdoctoral researcher in my group. So he used the same enzyme, this xylanase that cleaves next to the uronic acids. It creates this, this is a mass spec profile, this forest of different oligosaccharides. And we were really intrigued by this peak here, this X2U, because this is very interesting. In other species, this X2U is not so abundant, but in aspen it is. And we were able to see that this motif with 2 xylose and 1 glucuronic acid and acetylation is quite abundant. And we also could see that it's actually clustered, like a block copolymer. So we think that there are stretches in aspen that they have this very interesting X2U, 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 X2U motif. The function is, we don't know, but you can think that having an acidic group can have a function in modulating the pH, specifically in some parts of the cell wall. But not only that, we could also find this consecutive glucuronic acid motif that I mentioned before for spruce, but it has never been reported for hardwoods yet. So this is also quite interesting, that maybe this motif is also conserved in hardwoods, in this particular case in aspen. So maybe soon there will be some gene that is being discovered that is capable of putting these consecutive glucuronic acids. And also acetylation is very interesting. We know that, again, xylem from hardwoods is acetylated. And we also know that from previous works that acetylated is normally also conserved, very well conserved in hardwoods, like normally in even positions, again, to keep this even pattern when it always faces in the same direction. But we could detect, specifically in aspen, that some of it is over-acetylated. Somehow there is a little bit more acetylation than what it should be to this pattern. And we think that that might be due to the role of different plant acetylesterases in the cell wall, because these samples were taken from very young aspen that it was cultivated in the greenhouse. So this is work that is being wrapped up right now, and I think what that tells us is that the pattern of substitution is much more complex than we once believed. We're also working with eucalyptus xylem, and eucalyptus xylem is very interesting because apart from this structure that I already mentioned with glucuronic acid and acetyls, it also has galactose in the structure. Well, I'm trying to see if I can put the pen here. There we go. So it has this galactose linked here, and somehow it seemed to be some confusion in the literature whether it was alpha or beta linked. So now we have demonstrated with beta galactosidase that is actually indeed beta linked, but we wanted to see if there was also some kind of order in the distribution of these galactoses in the molecular structure of eucalyptus. And this is work done by Emilia, who is a PhD student in my group. So when we try to do the same procedure, using this fantastic enzyme, this GH30 that cleaves. Then we got this profile, but we could not see this galactosylated motif, although we know from the sugar composition that it's there. So we were very confused, we were very intrigued. Now we realize that actually it's a very small peak here, which is very, very interesting. But then we used other enzymes that are a bit more tolerant to substitutions, and then we can see it here. So we believe that this galactose now is somehow present in very cluster domains, or maybe in specific xylem populations. So we still don't know that yet. But I think it's quite interesting that this galactose seems to be maybe clustered and contributing to a specific function in eucalyptus. And then just a couple of slides about mannans. So we know that also mannans are very important hemicelluloses in plant cell walls. The problem of working with mannans with mass spec is that in glucomannans, we have three types of sugars. We have glucose and mannose in the backbone, and we have galactose as side chains. Unfortunately, the three of them, glucose, mannose, and galactose, have the same molar mass. That means that when we analyze them with mass spec, it's very difficult to separate them. It's basically impossible. They have the same molar mass. So that's quite tricky. But what we did in this study a couple of years ago is that we could isolate different populations of mannan from spruce, and we could separate these types also based on their fine structure. So I identified one, what we call accessible mannan, that has a bit higher degree of acetylation, around 0.3, 0.4, and it has a very low glucosylation and galactosylation. So here you can see the pattern of this mannan. And then we could find another type of mannan, which is what we call recalcitrant mannan, that seems to be very tightly interacting with, we believe it's actually cellulose fibers, that has less acetylation, but much more galactose and much more mannose. And using, again, chromatography and mass spectrometry, we could identify that somehow it seems that the presence of this galactose and glucose seems to be also quite well regulated, so in even positions. So then we went back to our collaborators working with molecular dynamic simulations, and what is fascinating with molecular dynamic simulations is that you can just have your cellular surface, you start building your different hemicellulose fine structures, and here we propose different backbones. We propose just a glucose backbone, a glucose mannose alternating backbone, a pure mannan backbone, and then some other backbones with not such an ordering of the mannose and glucose positions. And then we could actually measure in the computer how much energy does it take to basically detach these oligosaccharides from the surface of the cellulose. So then we could see that basically the fully glucose backbone is the strongest, but also the mannose glucose is basically has very similar interactions, while this mannose backbone somehow has less interaction. And then we could see that actually having this mannose in the backbone doesn't allow a full alignment and full interaction with the cellulose surfaces. So that's why we think that the interactions are a bit less stronger. And based on these studies, we could identify different populations in the cell wall. So a more, let's say, accessible mannan here with more acetylation and less in glucose, and a much more recalcitrant mannan with this even pattern glucose mannose that seems to be very highly attached to the surface of the cellulose. So yes, so and just last part on this presentation, I wanted to mention that we did a study trying to understand how different hemicelluloses interact with cellulose. We used the bacterial cellulose model as a model for that. We incorporated different wood hemicelluloses, xylans and mannans from birch and from spruce. And the idea was here to see if adding a specific hemicellulose would modulate the interactions on the biomechanical properties of these hemicelluloses. And indeed, what we found was quite fascinating, that the xylans really improve the tension properties, so the extensibility of the cellulose hydrogels, where mannans really helped in the compression properties. So we think that basically this very fine structure of the hemicellulose is being able to adopt both rigid conformations when they are adsorbed onto the cellulose surface, but also flexible when they are basically adsorbed in the different layers, can really modulate the interaction between microfibrils and also these biomechanical properties, both in compression and in extension. And basically, we're using this fundamental discovery that we found now to try to modify fiber surfaces using hemicelluloses as anchors. So just as a brief summary of this first part of the presentation, I hope I convey the message that hemicelluloses have really well-defined structures. It's much more complex than we once believed, and that they has a big impact on the way they basically interact with cellulose and lignin in plant cell walls. And as I already highlighted before, I think it's very important that we have much more collaboration between plant biologists and material scientists, because I think we have a lot of potential to really unlock the benefits of plant polysaccharides for material applications. And now in the second part of this, in the last 15 minutes that I have, 15, 20 minutes, I wanted to shift the attention from wood and from fundamental plant cell wall studies to cereals and to more food-based applications. So my other half of the group has been working a lot on using cereal cell walls as a new source of novel food ingredients. And this is fundamental cereal cell wall chemistry to start with. So we're looking at, for example, side streams from cereal production. Here you have a description of the wheat kernel, the cereal grain. Normally, the endosperm is what is used to make flour, and the bran is normally used as a side stream for animal feed. So what we're trying to do is to utilize this wheat bran to extract functional polysaccharides, hemicelluloses, dietary fibers, and to use them as functional food ingredients. Here, the challenge, as I will talk now, is really, again, the very tight supramolecular architecture of these cereal cell walls and the recalcitrants, which really hinders the isolation. So we're very interested, again, in hemicelluloses, in arabinose islands. This is again the molecular cartoon of the arabinose island in cereal cell walls. What is very interesting is that you have these ferulic acids covalently bound to the arabinose that are basically responsible for this cross-linking of the cell walls, these ferulic bridges. But not only there, they can cross-link to lignin here, they can cross-link to proteins. Then you have the cellulose microfibrils, you have the beta-glucans, you have the pectins. So basically, this is the architecture of the cell walls that we need to work with when we want to extract these arabinose islands and these different, to fractionate these different polysaccharides from this insoluble fraction. So traditionally, as you know, as I was mentioning before, people normally use alkali conditions that can cleave these ester bonds between the arabinose and the ferulic acid. And then the problem with this is that you somehow lose these ferulic acid moieties in the polysaccharides. What we have done instead is to propose this subcritical water extraction. It's water at high pressure and high temperature. And what we can do with this process is basically to extract these arabinose islands, keeping the ferulic acid bound to the polysaccharide core, basically not cleaving these ester bonds between the ferulic acid and the arabinose. We've studied this process quite well in detail. We know that, for example, if we start from wheat bran, which is the substrate that we have worked the most in the last years, normally when you run the kinetics of the extraction process, normally in the beginning you start extracting beta-glucans. You can see them in blue here, and the more the extraction goes, then these arabinose islands become much more solubilized. So, and we increase the purity of these arabinose islands. We know that also we can tune the arabinose to xylose ratio of the arabinose islands that we extract. We normally get arabinose islands with relatively low arabinose to xylose ratio. But as I was mentioning before, what is important is that we can keep this ferulic acid covalently bound to the arabinose island with around 1%, like 10 milligrams per gram of arabinose island. So this is quite important because we have demonstrated both in vitro that these arabinose islands are the ones that confer antioxidant and radical scavenging properties to these arabinose islands. And I will give you some examples later. Basically, you can apply this subcritical water process to any cereal or plant biomass. We've been doing that for wheat bran, from rye bran, from barley, from corn. And we have learned that basically adjusting the pH, the time, the temperature, we can really tune the substitution of the different arabinose islands that we extract. And I here put a cartoon of corn arabinose island because I find it very fascinating. It's a very complex island with a lot of side chains and decorations. And then again, fine-tuning the molecular structure, fine-tuning the conditions of the extraction. You can get corn islands with more or less substitutions and molecular weight. So what we've done in this further work is try to see how can we use these arabinose islands from cereals for different applications. And this was a work done by Cecile, by a PhD student. And she was working mainly with two types of arabinose islands from cereals because they have, as I was mentioning before, different properties. We were working with wheat arabinose island, which is somehow low substituted. It has a arabinose to xylose ratio of around 3 to 5. Quite respectable molecular weight, 100 kilodaltons from the bran. But we have also been working with corn, which is a bit smaller in molecular weight, but it has much higher ferulic acid content, like around 50 milligrams per gram, and it has this very nice branched architecture, a lot of decorations and very soluble, which is actually quite nice. So in the first study that Cecile did, we said, can we make films based on these wheat bran arabinose islands from wheat bran? We can. And what is really interesting, apart from the mechanical properties and the thermal properties, is that when we actually measured the antioxidant, the radical scavenging properties of the films, so we compared our arabinose islands with ferulic acid bound, and we compared it with a reference with arabinose island which does not have ferulic acid, but we added it as an external additive, as free, with the same amount as the bound one. We found that actually the antioxidant properties of the bound one was higher than of the free ferulic acid. So there seems to be important for these antioxidant properties that the ferulic acid is somehow bound to the arabinose island. We also been working with enzymatic cross-linking of these arabinose islands. In this study, we used lacase, which is an oxidative enzyme, which is able to basically oxidize and create a radical in the ferulic acid. You can see it here. And this radical basically is able to cross-link the arabinose islands again. This is a very similar process what happens in the cell wall when serial cell walls are constituted. So we call this process biomimetic. And when we did that, we could measure using different biochemical analysis that indeed the radical coupling was happening. And we could also identify what was the nature of these diferuloid bridges from a biochemical perspective. But what was interesting is that of course, we wanted to use these cross-linked arabinose islands for different gels. So we were very interested in monitoring the rheological properties of these arabinose island gels. So you can see here, we tried the soluble arabinose island. Of course, that didn't have very big viscosity. When we cross-linked it with the enzyme, with the lacase, we could see that it changed the rheological properties. It just basically increased the viscosity a little bit. And then when we dried this, when we freeze-dried and we regenerated it with water, we could see that the rheological properties and the viscosity really increased a lot. So we were very intrigued about this. So what was happening when we cross-link and when we dry and when we regenerate. And then we did wide-angle X-ray scattering. Here you can see the profiles. This is from the solubilized arabinose island. A little bit of order. I think this is because it's really low substituted arabinose island. When we cross-linked it with the enzyme, we somehow doubled the degree of crystallinity. And then when we dry it and regenerate, somehow it didn't increase so much. So we think that when we cross-link with the lacase, we somehow are promoting these backbone interactions here that I am marking with the cursor between the unsubstituted parts of the xylan. But then this still didn't explain the increase in rheological properties upon drying and regenerating again. And then we went to max 4. We did some small angle X-ray scattering, and I think Leo will talk about more in his presentation. And what we could see from these studies is that actually it was the network architecture that changed when we were when we are drying and regenerating. So not so much at the level of the individual chains, but at the level of the network structure. So that was the explanation that we could see for these interesting rheological properties of these gels. We worked as well with corn. As I was mentioning before, we were very interested in corn because here we don't have any unsubstituted backbone. We have a lot of side chains. We have also a higher content of ferulic acid. So this corn is very nice because you basically you add the lacase and it really gels very nicely. In this study, we investigated two different enzymes, one lacase, and you can see here, this is the way, or this is the time scale of the gelation. This is the decrease of ferulic acid upon gelation. You can see here it takes around 1 to 4 hours for the reaction to occur. But with the peroxidase, which is the other enzyme that we tested, it just basically after 5 minutes. You add the enzyme, you add H2O2, water peroxide, and it really forms the gel immediately. We were able to study in situ the gelation in the rheometer, and again we could see the different behavior. This is the lag case. You have the lag time, and then you can see here the increase in the storage modulus in the rheometer. While with the peroxidase, it was immediate. You know, you couldn't even measure it in the first times. And we could also see here, you can see some cryo-SEM pictures of the gels. That also this very quick and very slow gelation also affects the porosity and the properties of the gels. So this was very interesting because this tells us that basically controlling the enzyme loading or the kinetics of the gelation process, we can tune the rheological properties of these gels. But not only that, so we were very interested in seeing if this ferulic acid, again, because we use the ferulic acid as a handle for the gelation. So if keeping this ferulic acid would have some kind of antioxidant and an influence on the bioactivity. So what we did was we had these gels. We seeded some in vitro some colon cells in the gels. We added some radicals, and we could measure in time how these radicals would be quenched in our gels. We compared with an alginate, a very standard polysaccharide hydrogel, and here you can see really how the intensity coming from the radicals is really different. So in the alginate, you can see that with different times and different dosing, you can still have the radicals, but in our films, they really get quenched. So now we're really excited because this gives us an in vitro proof of concept that somehow we would be able to eat this, or we would be able to use these gels as, for example, encapsulator of probiotics or whatever you can think of, if they reach the colon, and they could have some antioxidant and radical scavenging effects in our colon. So we're exploring with collaborators now how to use them in a biomedical context. And also now we're very interested in understanding what's the scale or what's the network organization of these gels. So we've been running different tests recently using neutron scattering. And here again, the interesting thing is to compare these two extremes I've always been mentioning. So the wheat bran arabinoxylan, when we have a possibility of backbone interactions, and the corn bran when we cannot have these backbone interactions, when the interactions are mediated by the side chains. And this is work that is being now analyzed. This is our team now in the UK, in the neutron source. So now we have a lot of data, and hopefully we can get some information about these gels very soon. And just as a final slide, now we will not only investigating the arabinoxylans alone. We have now different projects trying to basically do a reassembly of the serial cell wall to see if we can basically do in the lab, in vitro, reconstitute the serial cell wall based on the individual components. We've been incorporating cellulose nanofibers with our hydrogels. We've been trying to cross-link as well plant proteins with our hydrogels. And maybe hopefully in a future presentation, I can share some of these results. Especially it looks very interesting for as you can imagine, for the cellulose and for the proteins. You can really tune the properties of these gels. So just as a summary of this second part, I hope I conveyed you that these arabinose islands, specifically the ferrulated ones, are very nice and interesting matrices to develop hydrogels and films for different food applications. That this enzymatic coupling, or basically enzymatic oxidation of the arabinose islands, enable very nice fine-tuning of these crosslinks that really control the properties of these hydrogels. The properties are not only depending on the chemical composition, but also on the physical interactions and on the kinetics of the enzymatic coupling, and also very importantly that the presence of this ferulic acid really confers very interesting radical scavenging and antioxidant properties to these hydrogels that we are exploring different both nutritional and biomedical potential applications. And again, this is the end of it. A lot of content, but I really hope you got a bit of a flavor of the different research, both from the fundamental and the applied perspective for the hemicelluloses. And of course, I really want to thank the team that is doing this. I try to mention each specific team member in the different parts. And yeah, I'm really happy. If you have any questions, please reach out to me. Thank you for the invitation.